So we've now got a dynamic duo coming up on stage, Melanie, Neeland and Rhiannon Werder, and I think it's really nice that Rhiannon doesn't actually sit within the IIGH theme, she sits in the stem cell medicine theme, but these two guys have hatched this fantastic collaboration, and we're going to hear about understanding infant lungs. Over to you guys.
Thank you very much, and hello everyone. So lung disease is a major burden for children. It spans chronic conditions such as wheezing, asthma and chronic cough, acute illnesses such as those caused by respiratory infection, as well as more rare disease like cystic fibrosis. A common feature across all of these conditions is a lack of effective treatments and also the lifelong impacts these conditions cause, including poor lung growth in childhood as well as poor lung function in adult life. So there's an understanding that new approaches are needed to improve the care delivered to children with respiratory conditions, and this is really the focus of our research program. So our program, as Steery mentioned, sits across two themes: infection immunity, global health and stem cell medicine. We're also nested within the Royal Children's Hospital, particularly the respiratory department, and we have a range of collaborators across the Parkville precinct. And in terms of the laboratory work, we're funded primarily by philanthropy, including the Chan Zuckerberg Initiative and the Novo Nordisk Foundation. So within our program, we recruit children as they present to the Royal Children's with the respiratory conditions listed on the screen there. We collect a range of biological samples. These include samples of the upper airway, so those from the nose, as well as the lower airway, so from the lung. We perform a range of different profiling techniques on these samples to understand the pathology of the disease. So this includes single-cell sequencing, proteomics, as well as stem cell models. And so really our goal is to integrate the data that we generate in the lab to identify disease endotypes or clinical subtypes of these conditions and identify new targets for early intervention. So this here is just an example of some of the data we can generate using single-cell sequencing, which is a powerful tool that has enabled us to develop the first cell atlas of the childhood lung. And so this data here is from samples collected from preschool children with cystic fibrosis. And in the figure, each dot represents a cell. And then for each cell, we get information on thousands of genes. And this allows us to understand how these cells are interacting with each other in the lung, their role in the pathophysiology of disease, as well as how and if they are responding to treatment. I also thought I'd present an example of how our pipeline may lead to a change in clinical care. So recently we applied our pipeline to children with chronic cough. So chronic cough is common in children, however there are limited treatment strategies. Management at the moment primarily relies on antibiotics, and this is despite evidence that antibiotics frequently fail to solve the problem and that disease recurrence is common. So what we actually showed in this group of children is that there are two disease endotypes or two subtypes of the disease. The first are children who have a hyperinflammatory environment in their lungs, and the second are children who have a non-inflammatory environment that actually isn't different to a healthy child. The children within the hyperinflammatory endotype have a particular type of inflammation that we call type 2 inflammation, and we know that this type of inflammation is treatable with already approved therapies that are used for children in other conditions. So this really does provide the first evidence that perhaps these treatments that are already available for children could be repurposed to treat children within this hyperinflammatory endotype, and that's something we're working on exploring further now. And so I'd like to hand over to Rhiannon, who will now talk to how she's applying stem cell models to develop new treatments for respiratory infection.
Thank you, Mel. So Mel's just beautifully explained to us how sampling the airways of children can help us to better understand respiratory disease. But one aspect of respiratory disease, which is respiratory infections, would be really hard to address in this way because the way that we sample the airways is quite invasive and we'd have to do it many times over the course of infection. So the alternative approach that we've come up with is to use stem cell-based models. Stem cells are cells that we can take from patients and reprogram them and then turn them into any cell in the body. In our case, we're turning them into the structural cells that line our airways, called epithelial cells, and the immune cells that live within our lungs. We can then combine these cells, along with samples taken from the airways of kids, into what we call mini lung models. We can then infect these models with viruses or bacteria that cause disease and better understand how these diseases start and how they progress. The other advantage of stem cell-based models is that we can make billions of these cells, which then allows us to apply them towards drug discovery pipelines to come up with better therapeutics. So to show you how these models look, these mini lungs, here I'm showing you stem cell-derived epithelial cells in red, and you can see that in green we have immune cells that Mel and her team took from the lungs of kids at the hospital. And you can see this really beautiful coexistence of these stem cell and immune cells living together in these mini lung models. So now we're trying to apply these models to come up with new therapeutics for pneumonia. So typically when we inhale a bug into our lungs, it will interact with our airways. And so this might make us a little bit sick, we might have a cold, but ultimately we can clear it. The problem is these same germs that cause a cold can migrate down into our lungs, into a region called the alveoli, and this is where it causes pneumonia. As Stiri mentioned, pneumonia is a leading cause of childhood mortality worldwide. Fortunately, though, we can try to better understand how pneumonia occurs by making stem cell-derived airway and alveolar cells. And we've been applying these to look at pathogens that we know can cause pneumonia. So this includes bacteria like Mycobacterium abscessus and viral infections like rhinovirus, RSV, and SARS-CoV-2, which is the virus that causes COVID-19. So the problem is, because we don't have treatments that target any of these bugs, kids are still presenting to the hospital with pneumonia. And our hope is that by better understanding these diseases, we'll be able to come up with new therapeutics to treat pneumonia. And finally, Mel and I would like to thank everyone who's been involved in these projects, our teams of teams, our collaborators, and also the children and parents who participate in the ARREST-CF, early air and breath studies.
Thanks, guys. Super cool work, I have to say. It's very, very exciting. Are there any questions in the audience? I guess one question for me, maybe Mel, is to just quickly ask you about that intriguing finding about the two different endotypes and what you think might be the sort of therapeutic pathway for the next steps.
Yeah, thank you. Great question. Yes, this is a quite exciting new finding that we have, this type 2 inflammation present in only about 50% of children with chronic cough. So what we're doing now is actually setting up in vitro models where we can stimulate these cells with existing therapies such as dupilumab, which we know targets is anti-IL-4 and IL-13, which is this type 2 inflammation, to see if it can restore that inflammation in vitro, and then that will then provide, I guess, the first steps to looking at this and applying this in a clinical setting.
Super cool. We've got one question from the audience, then we'll move on.
This question's for you. Can your organoid model transfer oxygen into the solution? And also, have you looked at the mechanical influence on the maturity of them?
Yeah, those are two great questions and intimately linked, in fact. So the only cell type in our lungs that we currently can't reproducibly make from stem cells are the type 1 alveolar epithelial cells, which are the ones that are responsible for gas exchange. And part of that is that they're the huge flat cells, and that's because their whole job is to diffuse oxygen into our blood. And so one way to potentially make those cells would be to stretch the cultures, the organoids, to actually get those cells to be produced, because we actually know that in development, the embryo breathing the amniotic fluid is what actually causes those cells to develop. So that's very much on the cards in the next couple of months.